So today I want to introduce our speaker. We have Dr. Joji Muramoto. He's an associate professor of cooperative extension and an associate cooperative extension organic agriculture specialist. He's with the UC Agriculture and Natural Resources and also with the Center for Agroecology and the Department of Environmental Sciences at UC Santa Cruz. Today, he's going to be speaking on integrated soil health management for plant health and the concept of One Health. So now I'm going to pass this over to Joji, so you can go ahead and share your slides.
All right. Good afternoon, everyone. Thank you for the introduction, Cheryl. My name is Joji Muramoto. Thank you for participating in this seminar. I'm very excited. And today I will talk about the framework my colleagues and I have proposed to facilitate the development of non-fumigant-based soil-borne disease and soil health management in agroecosystems. So today's outline, just as an introduction, I'm going to talk about anaerobic soil disinfestation as a biological alternative for soil fumigant for soil-borne disease management in organic strawberries. And just to tell you where did I come from, kind of introduction to the main topic. Then talk about the main things, integrated soil health management for plant health and One Health. Okay, well, let's get started. So I came to UC Santa Cruz in 1996 as a visiting scholar, but I started to work on organic strawberries right away. But one of the main challenges is the soil-borne disease management in organic strawberries. And then this picture shows the Versium wilt caused by Versium dariae. This pathogen and this disease is very difficult to manage because this pathogen can be hosted more than 400 plant species, including lettuce, tomatoes, potatoes, cauliflower, artichokes, apple, cotton, and strawberry is one of the most sensitive one, and some weeds as well. It also makes it difficult because it can survive for several years without host plants. So it requires to rotate in land out of host crops for 5 years or more. So no other tools were available at that time. Indeed, our UCSC organic farm, we have organic farm on campus, and we had outbreak of this disease, Versium wilt, in 2001 and 2002, 2 years in a row on our field. Regardless of 7-year crop rotation, we planted strawberry 1 year and crop different crops for 6 years and came back to strawberries. But we had this because our farm is very diverse, small-scale farms and growing 40 or more different crops annually. And it's very difficult to avoid host crops like lettuce and tomatoes. Ended up has this kind of outbreak. And being organic, and we cannot fumigate it, and so we're kind of stuck. Unfortunately, coincidentally, Dr. Ariana van Broeken from the Netherlands, a very good friend of the then director of Center for Agroecology, Carol Shannon, came over to the farm and encouraged us to try newly developed method called biological soil disinfestation. And then later we found the same similar principles method was independently developed in Japan. And then we started to learn from both approaches and trying to optimize for California strawberry conditions. And then we renamed it as anaerobic soil disinfestation, in short ASD. The principle of this approach is acid fermentation in anaerobic soil. To implement, there are 4 steps. First, incorporate labile carbon sources to feed the native microbes. We don't add any specific microbes, but soil has abundant microbes there. So we just trying to feed them with easily decomposable carbon source, such as rice bran or wheat bran. I'm going to talk about more in a minute. And then moisten this amended soil and cover by plastic mulch to limit the oxygen supply and gas exchange. Under the plastic cover, we're trying to maintain the fermentation process, which create unfavorable conditions for fungal pathogens, and keep it for 3 weeks or so. For ASD, there are 3 important factors affecting the intensity of anaerobic condition under the top soil. temperature, water, and carbon source type and rate. Those determines the intensity of anaerobic conditions. And during the fermentation process, byproduct such as organic acids, volatiles, and microbial community shift, and also toxic ions such as Fe2 plus and manganese 2 plus are created. And all those things work together to suppress fungal pathogens. This chart shows a very important work by Shashika at Cal Poly San Luis Obispo now, Strawberry Center. During her PhD work, she monitored what's going on in terms of metabolomes and microbiomes changes during the ASD process. So as you see here, oxygen and sugars are decreased in the first early stage from aerobic to facultative anaerobic phase. And lactic acids and CO2 increased. And then as time goes by, anaerobic conditions intensified. And then organic acids and very later stage methyl sulfides are created. Also, anaerobic microbes, fumicutes and Clostridium increased during the process. So those changes combined effect suppress fungal pathogens. After several years of trials, we optimized this approach to California strawberry production systems. So we use rice bran from Northern California, or recently we found wheat middlings works as well, and much more cheaper than rice bran. Apply at 6 to 9 tons per acre rate, broadcast it, and incorporate it into the soil, and list beds as usual, and cover with plastic mulch, and of course, and drip tapes too. And then drip irrigate total 1 to 2 acre inches. Those are standard strawberry practice. And then the amount of water is equivalent to the bed fumigation. And leave for 3 weeks or so. And then it is highly recommended to monitor soil EH redox potential, which is the indicator of anaerobic conditions during the 3-week period. And then add water if soil EH increases too early. And this approach works pretty well for Versium dariae and Macrophyllina too, but not for Fusarium oxysporum fragaria. This pathogen is very different. And then, being saprophyte, they can use dead organic materials, not just living, roots, but dead materials such as crop residues or rice bran. They love actually rice bran or wheat bran for multiply themselves. So we got to be very careful, especially under lower temperatures. This picture shows the example of one of those things. UTC untreated control. This is a field highly infested with the Fusarium oxysporum fragaria. So plants are dying here on the left side. And if you do ASD fall treatment, which makes things worse, more plants are dead. But if you do summer ASD, that works. So we add rice bran to feed the bacteria to create fermentation process. But a bacteria is more active in the higher temperature. And to compete with Fusarium, higher temperature is necessary. Now, our experiment shows more than 460 hours above 86 Fahrenheit at 8-inch soil depth is necessary to control this pathogen in coastal California. So it's kind of difficult to manage at this point. So if you have a Fusarium infested field, it's safe not to use ASD or rice bran. If you don't have it, you can use it. Another example, this is for Macrophyllina passerina, charcoal rot. The organic growers field far right has a very devastating conditions. But by using ASD with rice bran 9 tons, we were able to double the crop yield. And growers were really happy about it. And rice bran, also wheat bran too, has a 2 to 2.5% nitrogen in it. And it works as a pre-plant slow-release fertilizers. And it works pretty well for strawberries. So the data shows the mineralization rate of rice bran under aerobic, like blue lines, or anaerobic, a 3-week period, then aerobic condition like ASD. Either way, like 20 to 25% of total nitrogen, which is mainly organic form in rice bran, is mineralized, becomes plant available form in 4 to 6 months. So it works pretty well for strawberries, the strawberries fertilizers. And many growers use now rice bran just as a pre-plant fertilizer, which is fine as long as they don't have diseases. So we did 10 to 11 replicated field trials across California between 2010 and 15, and with using rice bran 9 tons ASD side-by-side comparison with untreated control and fumigated control. And the average relative marketable yield compared to fumigant, which is 100, at ASD plot is 99. So almost identical to fumigated control. And untreated control at 81. And if you add one more trial, highly infested with Fusarium, the average of relative yield of ASD decreased to 95, but still statistically similar to fumigated control. So with those kind of data, growers actually adopted to use rice bran-based ASD in 2011. We started just 1 acre in that year, but as of 2022, approximately 2,500 acres fields are treated by rice bran. We don't know how much of those are using ASD seriously, but mainly rice bran, other fertilizers. And then mostly in organic fields, which makes sense, even at organic fields, if you can get the higher yields like conventional and with price premium, why not, right? And then, but yet still, it's only 4 to 5% of California total strawberry acreage. So it still have limited in terms of adaptation. Now, ASD research has been not just in California, but across the 11 states at least, and 10 or 11 other countries for strawberries and vegetables and tree nuts and fruits and nurseries, like high-value crops, been the research and partially adapted. So future ASD research, we need to increase the efficacy of ASD to control Fusarium wilt at lower soil temperatures. We are running some experiments for that. And the greenhouse gas emission evaluation in California strawberry systems. There's data from Florida system indicating the N2O and CO2 emission was similar to fumigation. But we need more data in California. And finding less expensive carbon sources for ASD. Again, as I mentioned, recently we found wheat bran is a little bit less expensive than rice bran, which is good news. And then we are trying to find other carbon sources that are more affordable as well. Okay, that's a kind of introduction, a long introduction. But now let's talk about today's main topic. I was trained as a soil scientist focusing on fertility management. But in organic systems, organic amendments also affect soil-borne diseases. And I've been fortunate to work with many plant pathologists to study fertility and soil-borne disease management simultaneously in organic strawberries in California for over 20 years. The soil health concept has become increasingly popular in recent years. And many methods to measure soil health through measurement of chemical, physical, and biological properties of soils have been developed. I'm very interested in soil health concept as a soil scientist, but it often lacks soil-borne pathogen analysis. Soil health assessment without evaluating soil-borne pathogen can be misleading. Because healthy soil, defined by currently available set of indicators, can produce unhealthy low-yield crops due to soil-borne disease. Many soil health management practices, such as crop rotation, cover cropping, conservation tillage, and organic amendment applications, can reduce the incidence of soil-borne diseases, but the effect vary depending on the biotic and abiotic factors. Further, many plant pathogens are plant-specific. So, crop-specific or agroecosystem-specific soil health management strategy must be developed. So, Cheryl can be posted the link to this paper, but we published a paper, Integrated Soil Health Management for Plant Health and One Health 2022. And a podcast summarizing our paper is also available. The link is on that screen as well. Now, the concept I discussed today is called Integrated Soil Health Management, ISHM. This concept applies the concept of integrated pest management, IPM, and agroecological transdisciplinary participatory approaches to soil-borne disease and soil health management. The merit of this concept is to offer a framework for developing and implementing a comprehensive site-specific biointensive soil health and soil-borne disease management strategy. To explain the concept, the history of soil-borne disease management in California strawberry is briefly reviewed as a case study and contrasted with the evolution of arthropod pest management to learn some lessons and future directions for soil-borne disease and soil health management. So let's talk about the history of soil-borne disease management in California strawberries. Chemical fumigation using methyl bromide mixed with chlorpicrin had been the main tool for controlling soil-borne diseases and weeds in California strawberries since 1960s. It works extremely well, allowing growers to plant strawberries back-to-back at large-scale monoculture production systems, and was a core technology for the $2 billion industry. However, methyl bromide turned out to be an ozone layer depleter. As you know, the ozone layer plays an important role in protecting us from UV light radiation and resulting skin cancer, which is the most common cancer in the United States, more than all other cancers combined. Methyl bromide was added to the Montreal Protocol in 1992 and phased out in use in California strawberry fruit production in 2016, after 11 years of critical use exemptions. So after methyl bromide phased out, in response, California strawberry growers increased the use of other fumigants, such as chlorpicrin and 1,3-dichlorpropane. You see that this trend in this chart. Methyl bromide silver line decreased, and chlorpicrin and 1,3-D use increased in California. However, these fumigants are less effective compared to methyl bromide. The brown area in the photo indicate the dead strawberry plants by Thyrium wilt, regardless of chlorpicrin fumigation. This kind of scenery is gradually increasing in the area, which is concerning. Moreover, since 2003, California Department of Pesticide Regulation has documented hundreds of acute illnesses caused by accidental fumigant exposure to agricultural workers, as well as people living near fumigated fields. As a result, the regulation of fumigant is increasingly stringent, making future availability of fumigants uncertain. In 2008, the California Strawberry Commission began the Farming Without Fumigant initiative. Since then, multiple non-fumigant alternatives have been developed and examined. As I discussed already, I have been working on biological alternatives, alternative called anaerobic soil disinfestation, ASD, for over 20 years. This approach is the most commonly widely adopted non-fumigant alternative in California, including like rice run application. But the adoption rate is still really low, and more options, alternatives is definitely needed. Meanwhile, organic strawberry production in California has been increasing. As of 2023, over 5,000 acres, representing 13% of total strawberry acreage in the state, are now organically produced without fumigation. Organic production with price premium has been the main non-fumigant-based strawberry production system in California. Lastly, recent development of the molecular approaches in detecting soil-borne pathogens are opening up the new ground. There are three major lethal soil-borne pathogens in California strawberries. Fusarium wilt caused by Fusarium dariae, Fusarium wilt caused by Fusarium oxysporum form a species of Fragaria, and charcoal rot caused by Macrophylla fasciolina. The pathogen identification is the first step of IPM approach, since each pathogen has different character. However, the traditional culture-based method take about 1 month to identify a pathogen, making this approach impractical for IPM approach. Now these rapid and accurate molecular method, quantitative method, allow us to scout soil-borne pathogens in timely manner and to use the diagnostic-based IPM approach in soil-borne disease management. Note that this kind of molecular approach is still in development. There are some pathogens that still not have economical threshold, or the molecular approach has not been developed well, but it's definitely pathologists are working hard and to make this available to everyone at the production level. So, comparing the history of soil-borne disease management in California strawberry with the evolution of arthropod pest management offers some lessons and directions for the future of soil-borne disease management. So let's take a look at it. Between 1950s and 1980s, during the heyday of chemical revolution, non-selective, highly toxic pesticides were widely used in arthropod pest management, to the tune of the motto, "The only good bug is the dead bug." Basically kill everything. In the 1970s, the concept of IPM was developed, recognizing good bugs as well as bad bugs exist. In the IPM system, transitioning to biointensive or prevention-based IPM, as well as redesigning of cropping system aimed at fostering plant and insect community and population dynamics that self-regulate regulated pest presence and damage. And more recently, the extinction of some arthropod species and the decline of honeybee colonies has made us aware of the benefits of arthropod biodiversity and pollinators, leading us to realize that without bugs, we might all be dead. In contrast, for soil-borne disease management, non-selective fumigants are still in use, and IPM approach is yet at its onset. This delay is partially due to the unique challenges associated with the soil-borne disease management, such as invisibility of pathogens, the lack of post-symptomatic treatments, limited availability of non-fumigant alternatives, and the complexity and diversity of soil ecosystem. This is a biggest challenge, I think. Today, however, soil-borne disease management is situated in an exciting time, discovering beneficial and commensal microbes in the soil, like arthropod pest management found beneficial and neutral insect. Recent study also suggests that the core microbes who connect pathogen and beneficials in the soil may play important role. Research on suppressive soil and soil memory, plant immunity and rhizosphere immunity, and improved crops nutrient uptake by mutualistic fungi, such as mycorrhizae, are extremely active. As we better understand the soil biome structures and functions and their relationships with plant health, indicators of beneficial soil microbes and soil microbial communities for a specific crop or an agroecosystem may be developed. European Union, EU, has one of the world's most stringent fumigant regulations and is leading in the development of the IPM approach for soil-borne disease management. They developed soil health strategy actions consisting of prevention, including certified seeds, sanitation, and weed control, monitoring, including soil sampling, bioassay, crop rotation, including frequency, sequence, green manure, resistant varieties, and additional measures, including grafting, biological control agents, biofumigation, ASD, organic amendments, sterilization, etc. We propose that integrate soil health management, ISHM, as a science and practice with social movement advocacy for non-toxic agriculture. ISHM may evolve similarly to IPM for arthropod pest management towards biointensive management, increasing prioritization of the role of beneficial organisms and redesigning cropping system and cultural practices that prevent soil-borne diseases and induce sustained soil and plant health. At the same time, ISHM is more than a simple application of integrated soil-borne disease management. It also encompasses soil's many other functions by improving overall soil health using transdisciplinary participatory approaches. The proposed ISHM system in this context consists of 4 components. First one is comprehensive soil health diagnostic system. Second, farmers' location-specific knowledge and adaptability. Third, a suite of soil health management practices. And fourth, decision support tools. Overall goal is to develop site-specific agroecosystem design and soil health management strategies. Now let's take a closer look at each component. First, a comprehensive soil health diagnostic system. This is created by integrating molecular approaches for quantifying pathogens, beneficials, and soil microbial indicators and their thresholds. With an existing soil health measurement system measuring physical, chemical, and biological soil properties for assessing soil's other functions, such as nutrient cycling, water retention, and carbon transformation. The diagnostic system will determine the disease potential both from the pathogen density in the soil relative to their economic threshold and the disease suppressiveness of the soil towards target pathogens evaluated by its biotic and abiotic properties. This information will then be integrated with farmers' location-specific knowledge and adaptability. Although often overlooked and underappreciated, farmers' location-specific knowledge gained from day-to-day fieldwork and observations and their adaptability to dynamic agroecosystems and climate change is central to ISHM. Integration of scientific data obtained by diagnostics and farmers' experiential location-specific knowledge can be synergistic. Dialogue between farmers and scientists centers farmers as an active player in examining, fine-tuning, and scaling out agroecological knowledge and practices. Such participatory and transdisciplinary approaches mobilize knowledge for social change and engage stakeholders in research. The third component is a suite of soil health management practices known to improve soil health. As seen in the EU program, various soil health management practices, including practices for prevention and enhancing disease suppression via general or specific suppressiveness, for example, applying organic amendments, cover cropping, crop rotation, using host resistance, are integrated to tailor a site-specific soil-borne disease and soil health management strategy. A more intensive approach, such as ASD and steaming, is applied on an as-needed basis, depending on the soil health diagnostic result. Lastly, decision support tools will assist growers in developing site-specific soil health management strategies based on their goals, knowledge, environmental conditions, available management practices, results of soil health diagnostics, and other factors. Roles of decision support tools can be substituted or enhanced by agricultural professionals, consultants, and/or farmer-to-farmer networks. ISHM is characterized as a location-specific and knowledge-intensive approach, contrasted with the location-general and chemical or input-intensive fumigation and industrial farming approach. However, the transition to knowledge-intensive systems can present significant obstacle for farmers. As it worked in biocontrol, social learning, as seen in farmer-to-farmer networks. As it worked in biocontrol, social learning has facilitated the implementation and extension of knowledge-intensive soil health management. Policies and extension activities that support such a process and the adoption of ISHM will be necessary for the greater engagement in the co-development of ISHM with and among stakeholders. Some of you might have heard of the novel concept of One Health. We found this concept while preparing the paper of this ISHM paper. It's very fascinating and potentially powerful concept, which was first proposed to connect health between animals, humans, and environment by veterinarians. It was developed around 2000 when bird flu emerged. Since then, it has been expanded to connect soil, plant, animal, human, and ecosystem health through the cycling of diverse microbiomes. You must have heard of nutrient cycling, but the concept here is microbial cycling, which is fascinating. More recent paper listed more than 40 soil microbiome functions that either directly or indirectly contribute to soil, plant, animal, and human health. After all, soil, plant, and human microbiomes are perhaps more interconnected than previously thought. So in our paper, we included how One Health concept and ISHM relate to each other. Now, one of the best ways to develop integrated health management practices or soil health management practices is the learning from successful organic growers. I'm going to show you two examples for that. First example is Park Farming Organics, Scott Bryan and Ulla Park. Scott Bryan and Ulla Park. 35 years old, emphasis on soil health, and then more than 1,000 acres, 560 hectares, and 10 to 15 crops rotation, all certified regenerative organic, and then they manage the fields using the system of 9 C's: cover crops, crop rotation, conservation tillage, crop residue, conserve inputs, control traffic, compost, greater cover, and crew care. And he found his processing tomato fields have less leafhopper damages compared to his surrounding conventional processing tomato fields. And it was interesting. And he talked about it at the Eco Farm. And researcher from UC Davis, Emily Godin, and her group interested in the phenomena and started to do research. What's going on? The finding was fascinating. They found that changes in leafhopper settling between organically and conventionally grown tomatoes are dependent on the increased plant resistance by the salicylic acid accumulation in plants mediated by rhizosphere microbial communities. which means health of the soil actually increase the plant resistant to insect pests. Who'd have thought about it? Another example here. This is a PhD work by Tim Bowles. Now he's a professor at UC Berkeley, agroecology professor. But this is his PhD work. It's called Tightly Coupled Plant Soil Nitrogen Cycling. And then he surveyed 13 organic Roma-type tomato fields on similar soil types in Yoro County and found 3 different nitrogen cycling patterns. Group 1 is low soil nitrate during the plant growing period, throughout the period, and lower plant nitrogen level and lower yield. So basically nitrogen deficient kind of field. And group 2 was the opposite. Relatively high soil nitrate throughout the growing season and high plant nitrogen percentage and high yield. Which this is that kind of typical. But the third group is interesting. The nitrate level in the soil are pretty low during the whole growing season. However, plant nitrogen was similar to the second group, and yield was actually similar or even higher than second group. So what's going on in the third group? This third group, they call it tightly coupled plant soil nitrogen cycling, was supported by rapid soil microbial nitrogen flow without accumulating a large soil nitrogen, inorganic nitrogen pool. So soil nitrate analysis indicate the size of the pool, but the pool size was small in the third group, but the nitrogen flow was fast enough to provide enough nitrogen to plants. And those fields, group number 3, had the highest total and labile soil carbon and nitrogen, and received organic input with a range of nitrogen variability. That seems to be a key practice. And then Louise Jackson, emeritus professor at UC Davis, she was an advisor of Tim Bowles for this study, and of course the co-author, told me later when I talked to her, amazing thing is that these growers develop these tightly coupled plant nitrogen, soil nitrogen cycling system from experiences. True. This system was not developed by scientists, but by organic growers. They came up with this system by their experiences. That's very important. So probably important role of agriculture science to learn from pioneer organic growers, document it and discover the principles and share with others. And this is especially important in developing sustainable organic management practices, since many organic practices are grassroots practices developed by organic growers on their particular farms and farms with particular abiotic conditions. So it's hard to, disseminate unless we know the principles, what's going on inside. So I believe that's one of the very important way to develop sustainable management practices. Now, further story goes on, and this Louise Jackson, Tim's advisor, did postdoc work at Berkeley about nitrogen cycling in natural systems, and found tightly coupled nitrogen flow cycling in California native grassland. So she was thinking about those things for years, and encountered with the organic farmers field. So applying, natural systems function in agroecosystem is a very important principle in agroecology, and this is a beautiful example. And these days, state of California trying to use natural-based solutions, right? That's the kind of keyword these days, but this is one of the very good examples for that. Now, back to ISHM. Integrated soil health management shows only the framework and directions. Future research must fill the contents, and the needs are many. Examples include utilizing mechanistic models in plants' soil microbiome functions, such as soil suppressiveness, plant immunity, nutrient uptake, and better chemical and biological characterization of organic amendments and crop residues, and their relationship with soil-borne disease suppressiveness. Increase efficacy of plant growth promoting microbes in soil-borne disease suppression and nutrient uptake in field conditions. Many studies done in pot greenhouse conditions, but we really need field conditions research. Development of crop cultivars with ability to modify their rhizosphere microbiome for their benefits. And lastly, facilitating social learning among growers and transdisciplinary collaboration of researchers, growers, farm workers, NGOs, industry, and policymakers. Now, CDFA, DPR, and Cal EPA and others published the Sustainable Pest Management Roadmap last year, in which they state the goal is by 2050, California has eliminated the use of priority pesticides by transitioning to sustainable pest management practices. So they haven't defined what priority pesticides are now just yet, but they're going to decide what they are. And most likely, fumigants are included in this because of the amount of use and toxicity. But if that's the case, 2050, next 25 years, is very important to work more on fumigant alternative. And so if you are the younger generations than mine, I'm close to retirement, but for younger folks, this is very important task for you guys. As John Lewis said, you may not have chosen the time, but the time has chosen us. So if you are not sure what to do for the next 25 years, this is something very important for you to do. So, and I hope ISHM will help you guide that process. With that, thank you very much for listening, and I'd be happy to answer any questions.
So the one question so far that I see in the chat was somebody noted 13% organic strawberries. Has the percentage of organic strawberries been declining?
Say it again, last part?
So has the percentage of organic strawberries been declining?
No, it's increasing. Yeah, I didn't put the recent number, but it's like 13%. So I don't think it's declining.
Okay, and then a question just came into the Q&A. What is the impact with regard to Phytophthora development in the anaerobic soil treatment?
Phytophthora can be treated by ASD, but requires a little bit higher temperature, like Fusarium. But there's an example that's been treated by ASD. If needed, I can send some references to you if you could email me.
Okay, and then here's another question that just came in. Any chance I could use ASD to disinfect Fusarium spores, and I'm not sure I'm going to say this right, from a sorghum-based potting mix in a greenhouse setting?
Okay. I guess sorghum moss, yeah, like peat moss, I think. Yeah. Yes, you can do it. And actually in Japan, those like hydroponic systems, rock wools and peat moss systems, they use ASD to control Fusarium disease for strawberries and other crops. They use a liquid carbon source like ethanol, very diluted ethanol, like a 3% ethanol as a carbon source, and create anaerobic condition in the control system, like benchtop systems, and control Fusarium. that is possible. But again, it requires high temperature, above 30 degrees, so above 86 degree Fahrenheit. So it can be controlled at the higher soil temperature conditions.
Somebody's asking, what is the cost for ASD per acre?
Yeah, cost for ASD, that's good question. With rice bran, it's like with depends on the amount of rice bran. Rice bran carbon source is the biggest expense for ASD. And then rice bran currently like 300, more than $300 per acre. So if you apply 6 tons, like a $2,000 to $3,000 per acre, which is similar to much more expensive than bed fumigation, I guess, but cheaper than broadcast fumigation. And then, but it can be reduced by using wheat bran, like 20, 30% less expensive.
Okay, thank you. And then what is your opinion on compost tea and or vermicompost for increasing soil health via the microbiome?
Yeah, that is kind of a fascinating area. I I don't know exactly what's the most recent, but to my knowledge, compost tea can be used for reducing some foliar diseases and so forth. But you know, the tricky part is there's so many variables there. Depends on the material, what kind of compost you use, and so forth. So I think more research is needed, and the same for the effect on soil microbiome. Of the vermicompost.
Okay, thank you. And so the next one is, do you know of research linking soil health or bacterial communities, crop yields and quality, and positive human health outcomes?
Yeah, that's again, it's very fascinating areas. Um, I don't know direct research to show that kind of thing, but you know, there's a research showing, you know, microbiome and plant quality, like nutrient dense things. It's kind of on the trend. So, you know, there's some papers published about comparing nutrient density or nutrient content between different farming practices. Uh, but those are the things that so far we know of. And then, you know, yeah, so different mechanisms, but still, I think need more studies to understand.
Okay, thank you. Do you feel these practices are applicable to a deeper rooting perennial crop?
Well, if you are talking about the ASD, yes, it's it's good to use a liquid carbon source like molasses or ethanol. Then it can go deeper. You can treat deeper, and it's been applied to like a tree crop nurseries, almonds and walnuts, and tomatoes, nematodes, where that nematodes reside in the deeper soil layers, have been treated by liquid-based ASD practices. So that's another application.
Okay, here's the next one. From your knowledge from Fusarium or Verticillium, what do you know about applying this knowledge to control weed seeds? Are there any examples of success control?
Weed seeds. Oh, to control by ASD, I guess.
It doesn't say, but I'm assuming.
Yeah, wheat can also be suppressed by ASD, especially again under warmer soil temperatures. You know, ASD has a kind of overlap with the biosolarization, you know, which requires much higher temperatures, and solarization requires like, you know, above 100 soil temperature at 6 inches depth or something. But if you reach that high temperature and even, you know, at SD level above 30 degrees Celsius, 86 Fahrenheit, uh, it can suppress some weeds. But the high temperature is required than normal ASD.
Okay, and then the next one. I noticed steam treatment on your list of soil treatments. Is steam treatment compatible with ASD?
Yes, Steve Fenimore is a professionals working on Steam for many years. And then it's totally different approach. And then right now, Steve Fenimore is developing, to my knowledge, the mobile steam machine to like a spot treatment, you know, based on like a soil test or crop disease history, you know the damaged area or highly infested area, and you can do a treatment specifically targeting that area using steam. But he's still making the machine right now, so at this point that is not commercially available for open field scale. But yeah, he's working on it.
Okay, thank you. Are we seeing ISHM principles applied in the cooperative extension programming by county agents and specialists?
Not yet. It's not there yet, but if you're interested in, I'd be happy to talk about it more. But yeah,
Okay, thank you. Thank you, Joji, again for presenting. We really appreciate you presenting today, taking the time away to do that for us.
yeah, my pleasure. Thank you so much for having me.
All right. And so thank you, everybody, for participating today. And we hope we will see you back at our webinar next month.
Okay, thank you very much.
Thank you so much.
Bye.
Bye.